Good afternoon, ladies and gentlemen. My name is Adam Gilmore, and I'm here representing Horiba Instruments Incorporated. And today, basically, what I want to focus on is some application of some recently improved fluorescence and UV-vis absorption technology for monitoring the precursors of the disinfection byproducts that are associated with natural organic matter present in drinking and waste and water reuse industry applications. So basically, my talk is sort of loosely organized around this outline, a little bit about the background of disinfection byproducts, what they are, where they come from, different types of water treatment facilities, their monitoring points, the types of intervals that are involved, EPA regulations associated with the monitoring. Then I want to focus on the instrument and analysis requirements, because here again the recent improvements and technology have brought us forward and facilitated quite a lot of new applications. And then talk, of course, about the advantages and challenges thereof of this instrumentation, and then focus on some of the analytical approaches for extracting quantitative and qualitative information from these instruments. And then, of course, a little bit about what we see as the future, the immediate future, the near-term future, and basically international standardization projects that are underway associated with this technology. As I mentioned before, the topic being disinfection byproducts, it has a lot to do with drinking water treatment. Everybody uses various types of halogenated disinfection types that we'll discuss in a little bit, but basically drinking water, especially those treatment plants that are using surface water resources, are particularly concerned about the issue. Wastewater treatment, and again, the interaction between wastewater and drinking water in terms of the fact that they often share the same surface water sources. Wastewater recycling and barrier injection, of course, this is an important topic in our West Coast facilities. Some interesting developments associated and new points of view to deal with the hydraulic fracturing and flowback water disposal, and of course various industrial processing applications. So as I mentioned before, there's a number of water treatment types and facilities that we're interested in. So there's environmental monitoring. These disinfection byproducts are associated with the leachates of the soil, so they accumulate in almost all bodies of water, as we'll explain. They're associated with wastewater treatment, drinking water treatment, various industrial processes, semiconductor and pharmaceutical industry, power plants, desalination plants. So there's a wide variety of industrial and municipal applications for chlorine and other types of disinfection that generate these disinfection byproducts. The major precursor that we are interested in, that we primarily measure, is known as chromophoric dissolved organic matter, and that's generally defined as a permeate through a 0.45 micron filter, and is typically sourced from the decomposition of plant and animal material, primarily lignin of plants, leaves, and barks and stem. So the humic and fulvic acids, protein components, and chlorophyll degradation products are all associated with and known to be somewhat associated with these precursors for these toxic disinfection byproducts. Primarily, we're talking about things like trihalomethanes, haloacetic acids, and of recent, like I said, concern is the brominated species, not as opposed to the chlorinated species. We'll get into a little bit about that background, and some of the topics there associated with the hydraulic fracturing water disposal, which has very high concentrations of bromine and chlorine. So how do DBPs form? And I apologize that this screen resolution is a little bit different than what I had shown before. But yeah, basically the precursors are in water, the humic and the fulvic acids, the proteins and bromide. and your disinfectants being chlorine, chloramine, chlorine dioxide, and ozone. So basically those interact together depending on the contact time, the DOM, which is our dissolved organic matter concentration, disinfectant concentration, the pH and the hardness, the temperature, to form these disinfection byproducts that are in most cases known carcinogens, which is why they're of such concern. And there's been, of course, over the years, EPA Disinfection Byproduct Rule 1 that's been established for quite some time, and just for most large water treatment facilities, Disinfection Byproduct 2 has come into play. But it's primarily aimed in terms of our precursors, and like I said before, the surface water treatment plants that are using conventional filtration, having to deal with their running annual average, the type of treatment techniques that they use, and the water characteristics and hardness are all part of the disinfection byproducts rule. And the Stage 2 rule, which came into effect last year in April, basically had the main objectives to reduce the peak disinfection byproduct levels at individual locations in the distribution system, keeping in mind that the concentrations tend to be highest, of course, where the contact time is the longest, that being the point with the longest residence time in the distribution system. And so, of course, there are various requirements in terms of the monitoring test and the monitoring frequency, and your, what time of year you have to make measurements, and various other parts of these rules that are taken into consideration. And I think the key thing is that to avoid fines and to avoid problems for not meeting the requirements. So in terms of a typical surface water treatment plant, again, the surface water can undergo very drastic variations in DOM concentration depending on snow melts, floods, seasonal periodic droughts and things that cause different concentrations. So knowing the concentration of these dissolved organic matters from the outset is very important in terms of process control monitoring for controlling the coagulants and the other polymers and things that get added to the raw surface water before it enters the coagulation and flocculation tanks and sediments out and then gets filtered. Then again, they need to be able to predict how much is left and to control the contact time for the particular CDOM concentration before and after treatment. And as I mentioned before, the distribution system and the concentrations in the distribution system are something of importance. So measuring the finished water and periodically monitoring the distribution stream is basically all part of the disinfection byproducts rule. As I mentioned, the fracking industry has recently come under a spotlight in terms of its potential role to play in water contamination and disinfection byproducts. Typically, the injection wells, they have to inject a very large volume of water, and a certain amount of that water comes back up under pressure, and that's known as flowback water. And as you can see here, typically the wells are much deeper than the surface water and the aquifers, so it's not normally contamination along this phase that people are concerned about. Rather, what they're concerned about is the fact that when the water comes back up after they inject it into the, after they induce the fractures, and they collect and store this water, it's how they dispose of this water that's of most concern to the disinfection byproducts. Of course, in many cases, they take the water to recycling facilities or wastewater treatment plants, and they inject this, with very high concentrations of the bromide and the chloride that play a role in these disinfection byproducts. And in many cases, as I mentioned before, the wastewater treatment plants oftentimes share the source water with the wastewater treatment. And other cases, they inject the flowback water through the well. But it's in particular concern, this type of downstream situation for the disinfection byproduct precursors and the associated high concentrations of bromide and chloride So there's, again, been a recent EPA study that's actually still ongoing in terms of assessing the ability of this bromide and brominated compounds in the fracturing wastewater to form these disinfection byproducts. So again, there's a number of other carcinogen concerns in the fracking water, including PAHs and high chloride concentration. So the technology that I wanted to describe here is actually a rapid improvement in the time it takes to make these measurements of the disinfection byproduct precursors. So what we actually do is we measure fluorescence and UV vis absorption simultaneously. And we get two pieces of information that are actually correlated together and have an important relationship. Primarily, we have the absorption and fluorescence spectra of everything that's organic and has a fluorescence nature. At the same time, we also get the absorbance spectra of everything, whether it fluoresces or not, as long as it is an absorbing light or scattering light in the sample. So the importance of having these two things simultaneously is that the absorbance of light directly influences the spectral accuracy of the system. So we have to correct for the amount of light that's absorbed in the system and how this affects the fluorescence spectrum. And so typically, basically, our optical bench here comprises a light source, a monochromator that scans the exciting light that the sample sees, and it's also the light that's we do a sample and a blank comparison, but there's an absorption transmission path and a reference detector that corrects for changes in the lamp intensity. So you can make very long-term studies. It's fully corrected for the lamp intensity and changes in the lamp intensity. And the real breakthrough in speed comes from being a system that incorporates a CCD to collect data up to 100 times faster. So previous scanning instruments were actually limited in terms of the utility for monitoring the disinfection byproducts on a continuous online monitoring system because the scans took 25 to 90 minutes. And here we can take the same information in just 15 seconds to 1 or 2 minutes, depending on the concentration. So some of the instrument requirements, of course, as I mentioned, are the simultaneous visible and UV vis absorption fluorescence. The key thing that most of the aspects of the instrument were actually defined around several interlaboratory comparisons, primarily USGS organized interlaboratory comparisons. And so the key to this new technology is to have fully NIST traceable correction for all aspects of the instrumental and sample-dependent spectral features. And as I'll explain, the most important one being in terms of the wide dynamic concentration from the natural changes in surface water concentration, these interfilter effects associated with the absorption of the fluorescence excitation and emission portions of the spectra. In addition to that, there have been recent breakthroughs in terms of being able to fully automate software for continuous collection, correction, and multivariate analysis using various techniques for the data analysis. And then again, also the several aspects to dealing with coupling the system with auto samplers, flow samples, systems, multi-position sample changers, remote operation. One of the key features of the new technology, of course, also because it uses a CCD, you can get extremely wide dynamic range. You can get very deep, high sensitivity ultraviolet detection, so down to 200 nanometers for the absorption spectra and the excitation emission spectra, which turns out to be very important for a number of the low molecular weight disinfection byproducts that are of concern. And then on the other hand, there's importance in terms of measuring in terms of the near infrared and the red regions for chlorophyll contamination. In many cases, the chlorine disinfection and other addition of activated carbon are meant to remove products that are associated with algal and cyanobacterial growth, so blue-green algae. So the system actually allows you to quantitate the chlorophyll and various other accessory pigments simultaneously. But again, here, the ability to do this primarily facilitates the identification of harmful toxic algal blooms. So again, these odor, taste, and toxic compounds associated with the algal toxins basically are typically targeted as a part of the disinfected and activated carbon treatment. And so again, monitoring these allows the treatment plants to predict, treat, and to reduce the bad taste and odor compounds, typically known as geosmin and MIBK, and other toxins. As you know, there's been a recent outbreak of the red tide that killed a number of manatees in South Florida. But that's the same type of process of being able to, monitor the surface water source for algal contamination is something that this technique also facilitates. So some of the advantages of this, again, is it's very rapid. It has the potential for continuous online monitoring. Again, there's a number of advantages in terms of its comparison with the total organic carbon meter and gas chromatograph, and that it's actually a truly online capability. And it has the advantage is that the fluorescent compounds that you're measuring are, basically the most highly correlated compounds with the disinfection byproducts. So there's, in terms of TOC, there are a number of compounds that generally are summed together that you don't actually have a qualitative identification of the individual species. And in the case of the fluorescents, there's no room for inorganic carbon interference. And most importantly, again, the speed issue comes into play because most treatment labs, as part of their organic analysis, have to do a dissolved organic carbon gel fractionation procedure, which can take days and weeks. The same thing for the trihalomethanes forming potential tests, which come as kits and again take a very long time to develop and evaluate. So the online and rapid seconds to minute response again is orders of magnitude faster and provides information much more rapidly than these other typical tests. And in many cases, customers or people can operate this material, this instrumentation in parallel with gas chromatograms for monitoring trihalomethane concentrations. And again, the instrument provides all the information for the specific UV-absorbance, which is part of the EPA TOC monitoring method for evaluating the disinfection byproduct organic fraction. So again, the organic fraction, dissolved organic carbon, and the specific UV absorption measurements are fully quantified by the instrumentation. Of course, there are challenges in terms of any type of online system, maintenance, calibration, data acquisition, analysis, grab sampling and storage, especially with algal contamination, I guess, fixing samples, flow through monitoring, filtration, and maintenance. But again, nothing that is insurmountable, and again, it's something that we're very looking forward to the future applications. Again, one of the key things to deal with the system in terms of interlaboratory comparison and international standardization has to do with the wavelength and photometric accuracy and the spectral shape, and dealing with all aspects of the instrumental measurement itself, including removing artifactual light scatter that are associated with overlap of excitation emission. Being that it's water, there's a Raman scattering line that has to be removed, and interfilter effect corrections, which I'll explain, and standardized normalization. So you calibrate the instrument on a day-to-day basis. So this basically illustrates the interlaboratory comparison validation test for the system, comparing to NIST traceable reference materials of fluorescence in the ultraviolet and the blue-green. So the line being, the red line being the actual instrument performance, and the allowed error bars from the NIST certificates are shown in the error bars there. And again, there's throughput monitoring, so you continuously can, or on a day-to-day basis, can monitor the throughput and validate the instrument signal and signal-to-noise ratio. And again, the both the fluorescence and the absorption come fully certified with the typical NIST photometric accuracy and wavelength accuracy sample test with holmium oxide for wavelength accuracy and potassium dichromate for photometric accuracy. There's also tests for stray light rejection. So in a continuous operation, basically the system would first evaluate a blank sample with low total organic carbon and record the water Raman and the Rayleigh scattering. Then it would, the sample would be flowed in through the sample cell, and then the blank contributions subtracted, and then Rayleigh masks that basically have been developed to conform to the USGS interlaboratory comparison are applied. So then you see here you have the full fluorescence excitation and emission profile of the sample. And again, the key thing is to correct for the absorption of the sample, because the fluorescence as a technique is really evaluated where the excitation beam path and the emission beam path overlap. And so as sample concentration changes, there's a probability if the concentration gets too high that a fluorescent molecule will absorb a photon. And if it absorbs the photon before it gets to the area where the fluorescence can be measured, basically it reduces the intensity of the spectrum. And by the same token, if there's a fluorescence excited here, it can be absorbed before it leaves the cuvette. So in this case, you have two potential sources of issues that had to be overcome to apply this technology. So correcting this primary and secondary interfilter effect basically allow us to deal with a very wide concentration range of samples in this particular application. So this being an example of the issues people had with fluorescence before, where we're plotting the fluorescence intensity as a function of concentration measured through the absorption. And fluorescence is well characterized by being linear only over a very narrow concentration range. And when we apply the interfilter effect, we restore linearity and NIST traceable spectral accuracy over a much wider, basically 2 to 3 orders of magnitude concentration difference. So in this case, we have the ability to evaluate concentrations, again, well within the realm of most natural surface water resources. And it's very easy to recognize also when concentrations are well out of range in terms of needing to helping the plant to decide when to choose from a groundwater source or another surface water source that has lower dissolved organic matter concentrations. So some of the applications here in terms of wastewater treatment, just to give what some of the raw data looks like: clarifier water, tertiary treated water, and the barrier water that gets injected into the to prevent seawater intrusion. So this is a typical plant type of operation. The surface water treatment comparing the raw and the finished water. So in terms of application of the multivariate analysis, we're wrapping up to one of the last stages of this, is that there's been hundreds of papers on the subject, and we're collaborating with one group of researchers at New Jersey Institute of Technology and Middlesex Water Drinking Treatment Plant. But basically what they've developed is a principal component regression model that allows them to accurately predict the trihalomethane forming potential and the dissolved organic carbon with very high correlations. And so basically they calibrated it with the laborious gel fractionation procedure for measuring the trihalomethanes, and came up with an extremely robust correlation. So basically through a simple spreadsheet operation, it's very easy to take the EEM and break it into the principal components and to accurately predict within, well below the T critical value, all 6 fractions of the gel fractionation procedure, the dissolved organic carbon, and the trihalomethane forming potential. Other tools of interest, of course, have to do with, there's a number of ways. You don't necessarily need the full EEM in many cases, but principal component analysis and regression are very useful. Classical least squares is something that you can actually calibrate with known compositions, so you can deconvolute your mixture as a function of the linear components of the standards. Parallel factor analysis is another key thing that I wanted to focus on here, because there's been shown to be very high correlations between the components that are extracted with parafac analysis. So you're familiar with the three-dimensional excitation and emission spectra that I've shown here several times, but there was a special three-dimensional multivariate tool that was developed to separate these, and basically it breaks down the spectrum into each individual sample into a number of components that have, you know, the three-way matrix definition in terms of excitation, emission, and sample. So then again, it breaks down the samples into individual components, and these components basically can then be calibrated and stored in libraries, and even, you know, transferred from laboratory to laboratory because the instrument calibration is very effective. So basically, this parafac tool has been shown to correlate very highly with a number of water contamination and disinfection byproduct applications. This being one example of oil in water, and again, the system also facilitates a number of oil and water applications with this method in terms of identifying oil by evaluating the EEM and synchronous profiles of the EEM. So you have the absorption and the fluorescence emission data. And this being an applied parafac model that breaks and shows the crude organic fraction here in blue compared to the polycyclic aromatic hydrocarbon. You see the very distinct finger-like anthracene structure here separates very nicely, so you can, you know, continuously look for polycyclic aromatic hydrocarbons in the samples in addition to the disinfection byproduct formation. And it just so happens that the parafac has been recently exploding in terms of the number of publications. And there's, you know, dozens, if not hundreds, of papers now that show that the major components that you can identify with the parafac correlate very highly with the dissolved organic carbon and trihalomethane forming potential. Okay, so just some of the applications as a review here. CDOM is disinfection byproduct precursors. There's also a lot of interest in terms of wastewater treatment and desalination for evaluating membrane fouling, micro and reverse osmosis membrane fouling. Semiconductor and pharmaceutical industry, the total organic carbon levels have to be monitored. Oil and grease contamination in refineries and mines. Oil fingerprinting. There's a number of new regulations being established in terms of identifying specific sources for bilge water discharge and leakage of and oil spills, product and reaction intermediates and chemical and pharmaceutical process control, monitoring water for malicious contamination, water for nanomaterial contamination, algal and chlorophyll, as I mentioned before. So one of the things I want to talk about that's another very exciting development is there's going to be a fully validated public database of excitation emission matrix data and parafac and absorption spectra, hosted by Professor Rasmus Bro of the Department of Food Science, who's actually the author of the PLS toolbox for MATLAB and a number of other multivariate packages. And we're sponsoring this as, again, being a public database to facilitate interlaboratory comparisons in terms of both research and applied instrumentation. So there'll be various levels of validation applied in terms of full NIST traceability, as well as just general interest EEM data for compounds people are interested in identifying. So there's also a number of standards tests that are being updated and revised to focus on the advantages of the new instrumentation, primarily, as I mentioned, oil and water analysis because of the new regulations and Coast Guard regulations, EPA and Coast Guard. There's a very interesting standard practice for identifying, dozens of chemicals in water by fluorescence spectroscopy that's under revision. EPA method for the total organic carbon and specific UV absorption, again, being of interest in terms of the added advantage for the new technology, and the chlorophyll methods, as I mentioned before. And last but not least, we foresee, the people being very concerned about nanomaterials. And most nanomaterials that are toxic tend to have a high photoluminescence quantum yield, like quantum dots and other types of quantum materials, where the size of the material is what determines the spectral peak as opposed to the composition primarily. But again, in terms of defining future regulations, we think that this will play an important role. And last but not least, I just wanted to point out, again, the ASTM documents that are being revised in terms of the chemical significance associated with the Coast Guard and the EPA, and the chemicals that were identified from the Safe Water Drinking Act. And also a similar type of guide is being developed in terms of all aspects of the CDOM in the ISO procedure. So I'll admit I did an experiment here. I ended my talk without the conventional thank you and question slides, because I read several cases that that's a good idea. So at this point, I'll welcome any questions, and thank you very much for your attention. Sure.
As far as online monitoring in a raw water source, how would turbidity or changes to flow rates affect an online system?
Well, with this particular type of instrumentation, there's generally a pre-filtering that's applied, much like in a typical UV 254 meter. So the same potential or almost exactly the same requirements of going through a 0.45 micron filter, and sedimentation and other types of pre-clarification would be required.
So would that be changed daily during calibration, or?
Well, pretty much at the same rate. From what I understand, our colleagues generally change about once a week. So they have like a pre-column filters that are pretty easy to change. Yes? Yes.
Do you think we'll ever get to having a database for membrane fouling where it's not just the DOC, right? There's a whole interaction with
different types of polymers and things, yes. We were actually collaborating with researchers at the Orange County Water District and West Basin along those lines. I think that, of course, yes, that's something that, an application that we'll be targeting as part of the database. But only up to the point where it has a fluorescence or an absorption signature, I think, that fits within the guidelines of the instrument. Harold.
All the drinking water and wastewater treatment in the Mississippi River Basin in Texas is having high levels of pharmaceutical and halogen.
Yes. Well, again, in terms of wastewater recycling and wastewater treatment and surface water, there's a very high sensitivity to a number of the personal care and pharmaceutical products of interest, and that's actually encompassed many of those within the framework of these ASTM documents, especially the one for chemical identification. So yes, we have, like I said, a number of customers actually looking at surface water treatment of the Mississippi River that are primarily concerned with, well, I think they're more concerned with problems associated with their UV reactors and fouling of membranes and things like that. But yes, in terms of wastewater treatment and the effectiveness of it, the PCPs are something that we can target with parts per trillion up to, you know, subparts per billion accuracy.
Are you able to distinguish between the contributions made by hydrofracking to the input of fluorinated compounds from hydrofracking and those that are just used at the plant itself? Which are different?
Well, typically, the flow back water and the produced water tend to have, will have a very unique, depending on the geological area, polycyclic aromatic hydrocarbon and oil fingerprint. So that's one thing. Like I said, the system is very popular for a number of forensic applications for identifying oil from ships, from different wells, from spills. So it's very effective even at crude oil, characterization. A number of times also they may be using tracer dyes that are specific to a given well that can help to identify things. But the real focus and the main concern of the hydraulic fracturing isn't so much the other chemicals per se as the high bromide and chloride concentrations that are getting into the surface source water, from what our point of view is. So we're primarily focusing on not the water that's going into the wastewater treatment plant, but what comes out, and how that interacts with the surface water dissolved organic matter. Okay, well, thank you very much.
Thank